Hello, I'm Alex. I'm a PhD student at the University of Glasgow, and today I'm going to describe how to use topic modeling to detect cellular crosstalk in single-cell RNA-seq. Over the past few years, single-cell RNA-seq has been gaining immense popularity, and it's been used to understand heterogeneity of cell populations, as well as to study processes through the use of pseudotime and trajectory inference. Another application of single-cell RNA-seq relates to understanding cell-to-cell interactions, as cell-to-cell interactions are vital for numerous biological processes, from differentiation and development to disease progression. To date, there are two main strategies for analyzing interactions. One relies on ligand and receptor databases. An example of such an approach is CellPhoneDB. While such strategies are very useful, they are limited to known biology, and curated resources of ligands and receptors exist predominantly for human data and mouse orthologs. An alternative to the ligand receptor-based approach is having access to physically interacting cells. Early last year, a paper was published that following tissue dissociation relies on staining for mutually exclusive markers and then sorting and sequencing the single positive and the double positive populations. This approach is known as sequencing of physically interacting cells, or PIC-seq for short. On the computational side of things, the single positive populations are clustered, then synthetic reference profiles are built, and in order to identify genes that change as a result of interaction, the expected and the actual expressions are compared. Here, we present an alternative to the approach described earlier. Our method, that's based on latent Dirichlet allocation, does not require prior clustering or generation of synthetic reference profiles. In our setting of LDA, cells are equivalent to documents, genes are equivalent to words, and a topic is essentially a group of covariant genes. Word frequencies correspond to gene counts. We begin by fitting our first LDA on a reference population. This reference population could be sorted cells or the cells before the interaction has occurred. Following what we obtain from our first LDA in terms of gene and topic probabilities, we fix some of the topics before we fit the second LDA on the interacting population. It's the result of the second LDA we use to generate a comprehensive ranking of genes that might be involved in interaction. The ranking is based on in how many cells a gene requires the contribution from the new topic to have its expression modeled. Genes that have been involved in interactions would require contributions from the new topics to model their expression. We validate our approach on a range of real and synthetic datasets. The first real dataset we use has been generated by the PIC-seq approach described earlier. We have two separate co-cultures of T cells and dendritic cells taken over 3 different time points, and we use that as a reference. Here on the left-hand side, the heat map shows the expression of the topics over our cell types. Using Mann-Whitney U-test, we determine which topics are cell type specific. For example, topic 12 appears to be specific to the dendritic cells, while topic 23 appears to be specific to the T cells. Looking at the genes with highest probabilities in those topics, we see genes that tend to be expressed commonly in one of those cell types over the other. We use the topics we learned from the reference and fix them before we fit a second LDA of co-culture between T cells and dendritic cells, again taken over 3 different time points. The heat map here shows some of the genes we identify to be changing as a result of interaction, as they require contribution from the new topics to model their expression. The heat map shows the expression of those genes over the 3 time points of the co-culture. The genes we identify, some of them overlap with the original publication of the PIC-seq paper. However, we also note some further candidates of interaction that could potentially be explored further. Some of the genes we identify are related to immune response, but we also note some genes that are adhesion molecules. This is another evidence of physical interaction between the cells. We also note that some of our new topics are potentially capturing a temporal pattern, as their expression increases after the 3rd time point. Examples can be seen on the heat map. The next dataset we use is a bone marrow dataset where the reference contains sorted cells. Our interacting population, again, is interacting doublets. On the heat map, we show some of the genes we've identified to be changing as a result of interaction. Once again, adhesion molecules are amongst the top ranking genes. While the two previously described datasets have been generated by technologies that allow us to capture physically interacting cells, this is not the case for this one. For this experiment, we use a dataset generated by 10x Chromium, a widely used sequencing platform. The dataset used here is a COVID sample where there is potential interacting population of T cells and macrophages. What's important to note here is that we don't really have access to a reference population. Our reference has been determined by using doublet finder on the sample. Cells identified as singlets have been used as reference, while the doublets in the sample represent our potential interacting population. What our LTA-based approach is able to capture is in fact some shift in the expression of some cytokines and chemokines, which might suggest interaction. What we think might be happening is that the doublets are actually a mix of technical and interacting ones. However, we cannot be certain due to the quality of our reference population, as no doublet tool achieves perfect sensitivity and specificity. As such, currently we recommend using our approach for determining genes that could be candidates of interaction to be used on datasets that are actually capturing interacting cells. And here to summarize, we proposed a method that does not require curated resources of ligand and receptors, prior clustering, or generation of synthetic reference profiles. This is the first application of LDA for detection of genes that could be changing as a result of interaction. We produce a comprehensive ranking of genes that could be changing as a result of interaction, comparable with the original publication, as well as identifying some further genes worth exploring. Thank you for your time, and I'm happy to take any questions.